Now, the autoclaves are used to kill all the microorganisms that we work with in the laboratory setting. And it does so by increasing the temperature to about 121 degrees Celsius, which is incredibly hot, and it also increases the pressure. Once the pressure increases to about 15 psi, then you have basically the breakdown of the microbial organisms cell through the process of pressure, and once that happens, we're able to kill the microorganisms. So the use of heat, gas, and chemicals, as well as radiation, are some common methods that are used to kill microorganisms through the process of sterilization. And sterilization has to take into account that there are some structures that microorganisms have that can withstand certain types of treatments. So things like spores, if we have an organism that produces endospores, we have to treat it differently than a regular organism that doesn't have them. If you're trying to kill a bacteria that contains endospores inside of a autoclave system, you essentially have to increase the temperature to higher than 121 degrees Celsius, and then increase the length of the autoclave time that's used in order to kill microorganisms. Sterilization is different from the process of disinfection because with disinfection, all we're simply doing is removing pathogens from a non-living object. And this is through either physical or chemical means. And an example of this is pasteurization of milk, where you simply are heating milk to a certain temperature to kill some of the microorganisms in it. Disinfectants are chemical substances that can be used to get rid of pathogens on inanimate objects. If we were doing a laboratory this summer, you would be using bleach to decon your workstations because we use bleach as a disinfectant to kill pathogens, so that way they can't get to areas where we don't want them. Anything that is used and is applied to human skin to get rid of microorganisms is referred to as an antiseptic. So antiseptics are safe to be used on the skin, people or on animals, but disinfectants are not. Disinfectants might be way more corrosive, such as bleach, and so high concentrations of a disinfectant when applied to the skin can cause a lot of damage to the human tissue. So antiseptics were invented because they were safe to be used on skin tissue. There's a lot of different agents that we use to control microbial growth. There's things called germicidal agents, biocidal agents, microbial agents, and other chemicals that help to get rid of microbes. What all these agents have in common is the fact that they do kill microbes. So when we talk about controlling the growth of bacteria, if you see cidal or cide in the name of a particular agent, know that that means that that agent is going to work by killing the microorganism. Okay, so not all agents that are used for decon purposes kill microbes. Some of them just simply inhibit the growth, so the bacteria will still grow, but it will no longer replicate. So it doesn't really kill on contact. Bactericidal agents are going to be chemicals that specifically kill bacteria, but they're not able to destroy bacterial endospores, which can be problematic because once those endospores come into contact with nutrients resources, then the endospores are going to flourish into bacterial again. So instead, you have different agents called sporicides, which are used to kill bacterial endospores. Fungus also produce spores, and so they require a different type of agent used to minimize the production of spores, so we call this fungicidal agents. There's also agents out there to kill algae as well as viruses, and those are referred to as algaecidal and virucidal agents. A microbistatic agent is a drug or chemical that will prevent the growth and reproduction of microorganisms. We can also call this bacteriostatic. They're both kind of similar to each other. However, bacteriostatic is an agent that specifically inhibits metabolism and reproduction of bacteria. Basically, they're essentially the same thing. It's just one goes after metabolism, which is bacteriostatic, and then with microbiostatic, you're going after growth and reproduction. A process that's used to kind of store microorganisms is called lyophilization. And what happens in a laboratory is that you take your microbial specimens and you dry them, and then after drying them, you freeze them. And this helps to preserve different items that are generated in the laboratory, such as foods, antibiotics, microorganisms, and then different biomaterials. Some other terms to know regarding this particular section is that of sepsis and antisepsis. Sepsis simply means that you have the presence of pathogens contaminating tissues or contaminating blood samples. The word asepsis means without, and sepsis, contaminants. So asepsis means the absence of pathogens. And then there's the term antisepsis, which is the prevention of infection. So, we like to use different methods to inhibit microbial growth, and heat happens to be one of the primary methods that is used to do this. Temperature and time are both factors that determine how effective heat will be for the process of sterilization. So there's this concept that's known as the thermal death point, and every species is going to have this, which is essentially the lowest temperature that is needed in order to kill all of the organisms in a standard pure culture at a certain time. So as I was talking about that autoclave, remember, we like to use the autoclave to kill bacteria. But if you're dealing with a bacteria that might be an extremothermophile and can withstand temperatures higher than 121 degrees Celsius, then you have to do some research and figure out what the thermal death point is for an organism that lives in temperatures higher than 121 degrees Celsius. And in doing so, this will help you to figure out the best time and the best temperature to use in order to control the microbe. You can use various types of heats as well. There's dry heat and moist heat. Dry heat is when you use either an oven or a flame or an incinerator to kill microorganisms, whereas moist heat is where you use boiling or an autoclave in order to kill bacteria. So, two different types of heat. And here is an example of dry heat being used in a laboratory setting, and we do this all the time in our laboratories. You use essentially a Bunsen burner flame, and you pass your inoculation loop that has contaminated bacterial specimens on it through the flame to burn them off, using dry heat as a sterilizing technique. Now, there's been a lot of advances in the sciences, and because Bunsen burners can be very dangerous working with in the laboratory, there's also some electrical heating devices that can be used nowadays as well. The autoclave system, I talked about this a little bit already, but a little bit more detail about it is that it basically is a pressure cooker that relies on steam and pressure to kill microorganisms that are placed into it. As pressure increases, as well as the temperature increases, the water inside the autoclave system will start to boil. And as that water starts to boil, it's going to generate steam, and the steam is what is the key factor that causes lysing of bacterial cells. So again, the autoclave will work at a pressure of 15 psi, and you want to operate that pressure at a temperature of 121.5 degrees Celsius for 20 minutes. In doing so, you'll be able to destroy microorganisms, bacterial endospores, as well as viruses. And to ensure that your autoclave system is working, we actually use something that's special that's called an autoclave tape. This autoclave tape, when you put it in the autoclave, is actually going to be beige with none of these black markings on it. But when you take this tape out of the autoclave and you affix this tape to a test tube culture, or you affix it to a test tube rack, or whatever you're putting in the autoclave, just put a piece of tape on it. And if you when you pull your product out of the autoclave system, if all of these bands are black, then that means that your autoclave system is working. If these bands aren't black, then you've got a problem, and your microorganisms are probably still living inside of whatever it was you were autoclaving. So that's one way to tell if the autoclave is working properly. Another way to tell is to use these biological indicators. These are indicators that contain an organism called B. Stereothermophilus. It's an organism that can essentially live a little bit longer at high temperatures. And so what you do is you place this inside of the autoclave system and you increase the temperature higher than 121 degrees Celsius. And then once you autoclave it, you take it out of the autoclave and you get a control. So your control is the vial that was not autoclaved, and then your experimental is the vial that was autoclaved. And then the next step is to incubate both vials at about 65 degrees Celsius for 48 hours. If you come back the next day and you see that your indicator has turned from purple to turbid, or purple to yellow, then that indicates that your bacteria is alive. And so if this was your bacterial culture that you autoclaved, you would know that your autoclave is not working because this B. Stereothermophilus didn't die like it was supposed to. So we use biological indicators to help us determine whether or not our autoclave system is working optimally. And some other methods that can be used to inhibit microbial growth, some of these we talked about already. Cold, like we talked about refrigeration. Microorganisms don't die in the cold. That's not always the case. But instead, they're not able to replicate and divide, and so they just become sessile. With desiccation, you simply have the removal of water from microorganisms. So the microorganisms become very, very dry, but they're still alive. So the cells are there. They're just not able to reproduce, so they can't grow and develop. We can also control microbial growth using radiation. UV light is a great denaturer of proteins. So we can use a UV lamp to reduce the number of microbes that are present in a certain area. If you wanted to sterilize your light, sterilize the air, you can use a UV lamp to reduce the number of microbes that are present. We can also use some less unconventional techniques to control microbes, such as the use of sonic waves. Ultrasonic waves are those that are used in hospitals and medical dental clinics, and these waves can be used to clean equipment. So the ultrasonic waves just get so powerful they cause the cells to lyse. We can also use filters to separate cells from microorganisms, to sterilize liquids and gases. So there's some medias out there that can't be autoclaved to ensure sterility, so we end up filtering the media. We can also have a gaseous atmosphere, which can be used to inhibit growth of microorganisms as well. So again, the presence of different types of gas can also influence growth. Chemical disinfection is kind of our go-to when it comes to controlling microbial growth, inhibiting the growth of pathogens. Bleach is kind of the primary agent that's used to destroy bacterial organisms. Disinfectants also have some factors that affect their usage. The amount of grit that needs to be cleaned can influence whether or not the disinfectant will work.